My name is Shaylia Stanley, and the focus of this presentation will be the effects of age and exercise on human skeletal muscle fibers. As part of the aging process, there is a reduction in skeletal muscle quality. This is in part due to fat or lipid infiltration, a decrease in muscle fiber size, and alterations in muscle protein expression. As a result of this, muscle contractile properties decrease, leading to functional limitations and disability. Previous studies have shown that exercise can improve skeletal muscle quality, but the underlying mechanisms behind this change are unclear. The objective of this study was to determine how skeletal muscle structure responds to different exercise programs in males and females. Each volunteer underwent a 16-week exercise program where resistance training, which consisted of low velocity and high load exercises, were performed in one leg, and power training, which consisted of high velocity and low load exercises, were performed in the other leg. Tissue samples were obtained via a muscle biopsy of the vastus lateralis muscle in the thigh by a physician. The samples were placed in optimal cutting temperature, compound, and frozen with liquid nitrogen. A cryostat was used to obtain 6 micrometer thick cross sections of skeletal muscle tissue. These samples were then mounted on a slide as shown here on the right. This image on your right here is a skeletal muscle cross-section stained with fluorescent antibodies. Each individual segment represents a single skeletal muscle fiber as shown here, as I'm circling. And the immunohistochemistry consisted of staining the cross-sections with fluorescent antibodies for myosin heavy chain proteins to determine fiber type. The most common isoforms found included MHC1, which are depicted in green here. And these are the slow contracting proteins. MHC2A fibers are faster contracting and are depicted in red here. Even faster contracting are MHC2A2X and are shown in purple here. The MHC2A2X is a hybrid of the MHC2A and the MHC2X, though MHC2X are hard to find. For analysis, I hand traced 12,576 fibers from 25 participants. Of these participants, 14 completed the training. For those who completed the training, 4 were male and 10 were female. This data represents the proportion of myosin heavy chain isoforms in the muscle cross sections. The open bar represents males pre-training. The light gray bar represents males post-training. The darker gray represents females pre-training, and the black bar represents females post-training. In resistance training, a training by sex relationship was found, meaning that within a specific exercise modality, males and females experienced opposite results. Men experienced a reduction in proportion for their slower contracting fibers, or their MHC1 shown here, where females experienced an increase with resistance training in the proportion of these fiber types. No other significant results were found in the other fiber types for resistance training, nor were there any significant results found for power training for each of the isoforms as well. Overall, the reduction in males' slower contractile fibers reverses what is typically observed as part of the aging process. This data represents the cross-sectional area of individual skeletal muscle fibers. In resistance training, there were training by sex effects observed for all 3 fiber types, where the male's fibers got smaller and females either increased or stayed the same. The same relationships were observed in power training in all of the muscle types, or the MHC isoforms. The increase in fiber size observed in females reverses what is typically observed in aging. I'm currently working on quantifying intramyocellular lipid content, or fat within the muscle fibers, of younger low fat and high fat and older low fat males and females. This sample consists of 8 young low fat, 10 young high fat, and 6 older low fat individuals. This image depicts the staining process here. This middle image is a muscle cross-section stained with oil red O to highlight the intramyocellular lipid content. And this is an image that has been analyzed. Overall, there were sex-specific responses to the different exercise programs. In older men, resistance training decreased the amount of slow contracting fibers, which counters what we typically observe in aging, where the opposite was observed in females. There was an increased amount of slow contracting fibers with resistance training. However, in older females, the fiber size increased with both resistance and power training, which counters what we typically observe in aging. But males had a decrease in fiber size with both training types, resistance and power. In the future, the hope is to continue this study once older volunteers are able to return to campus and we can further assess the functional benefits of these findings. Lastly, I would like to thank the Muscle Biology Laboratory and Dr. Mark Miller for the ability to do this project, as well as his guidance and advice. I would like to thank the University of Vermont for slicing, staining, and imaging the tissue for the immunohistochemistry data, and the Institute of Applied Life Sciences at UMass for access to the facilities to prepare samples and image for the oil red O and lipid staining. I would like to thank the Commonwealth Honors College and NIH for funding these projects. Thank you.